Okay. I'm going to continue the discussions related to ANDA approval and major deficiencies, and I'm going to use a case study with topical products for the purposes of this presentation today. So the learning objectives for us is to understand the ANDA landscape for topical products applied to the skin, specifically what kind of approaches are being utilized to support an evaluation of BE, and then to understand what the challenges are and how do we resolve those challenges so we can eventually get to first cycle approvals. So the outline of my presentation has a brief recap of the BE approaches that we currently recommend within product-specific guidances for topical products. And then the second part of the presentation talks about landscape analysis of ANDAs that have been submitted to the agency since GDUFA II, so FY18. And then, of course, the challenges and opportunities. So a brief recap from the presentation that Dr. Kelchen provided yesterday morning. There are predominantly 4 types of BE studies that are currently recommended within the guidances for topical products. We have the comparative clinical endpoint BE studies. These can be used by all products irrespective of the formulation. Then we have the vasoconstrictor studies. These are typically used by corticosteroid products. We have situations where a waiver of in vivo BE studies may be applicable, typically for topical solutions where bioavailability may be self-evident. And lastly, the characterization-based approaches that have been developed within the scope of the GDUFA Regulatory Science and Research Program. So let's now look at some data. So I'm going to take some time on the slide to orient you to this slide because all of my subsequent slides are structured the same way. This is a summary of the submitted ANDAs that we have received since FY18. And the way the data is presented here is data for a given fiscal year is normalized to the total number of ANDAs received in that fiscal year. For the purposes of this presentation, approved ANDAs indicate any ANDA that has been approved or tentatively approved. Pending ANDAs indicate any ANDA that is in pending status, either in a CR status with the applicant or in pending status with the agency, and of course, withdrawn ANDAs. So overall, if we look at all of the data that we have since FY18 till FY24, the data is as of June 30th of 2024, we see about 55 to 57% of the ANDAs that we have received during this time for topical products applied to the skin have already been approved. Now let's take a look at what kind of approaches were used to support BE in these submitted ANDAs. So if we focus on the left of the screen here, we see the characterization-based approaches. And since the publication of the guidances related to these characterization-based approaches in the FY18-19 timeframe, we have seen a reasonable increase in the number of ANDAs that have utilized characterization-based approaches to support BE. And the number that I would like for you to keep in mind is this FY2021 number, where about 70% of the ANDAs used the characterization-based approach. We have also seen consistent submissions for products which have used clinical endpoint BE studies, VC studies when appropriate, and of course waivers when appropriate. Now let's look at what has been approved. So when we look at approvals, if we focus on the characterization-based approach first, remember the number 70% that I mentioned for FY21, we see that about 35 to 40% of the ANDAs have been already approved. So overall, we see approximately 40% of the ANDAs that have used characterization-based approaches have been approved until now. We also see consistent approvals for the clinical endpoint and the VC studies, but I'm going to talk about those in a little bit more detail when I talk about pending ANDAs. If we take a closer look at the approaches that used characterization-based approaches, we see that a substantial number of those products have successfully completed IVRT studies, and also some of those products have successfully completed IVPT studies. So I just wanted to note that before we look at the challenges we face in this area. So now looking at pending ANDAs. As we look at pending ANDAs in trend with the submissions where we saw an increase in utilization of the characterization-based approaches, we also see an increase in the number of pending ANDAs when a characterization-based approach has been used, and I'll delve into this a little bit more on subsequent slides. For the clinical endpoint studies, VC studies, and waivers, we do not see as many pending ANDAs, but I'll talk about overall some of the challenges that we do observe. So for the overall pending ANDAs, if we were to break them down about the challenges, we see about 50% of these products still have outstanding challenges related to bioequivalence, pharmaceutical equivalence, and in some cases also labeling, for example. But the other 50% of the ANDAs where bioequivalence have been found to be adequate, there are outstanding challenges related to pharmaceutical quality. They could be related to either drug substance or drug product, and also related to manufacturing and process and the facility. A limited number of cases where we have labeling challenges. So one thing that I wanted to note here is, while the pre-NDAs that I have been involved in have predominantly focused on the BE approaches, because this is new in the area of topical products, there may be some room for discussing pharmaceutical quality-related challenges in the pre-NDA space when those are appropriate as well, so that we can resolve those challenges and get to that first cycle approval. Delving down a little bit into these characterization pending ANDAs that utilize characterization-based approach, we see about a quarter of these ANDAs, quarter to 30% of these ANDAs have adequate IVRT study. For the remaining ones, while this segment looks big, I will note that the majority of the challenges that we observe with IVRT are primarily related to IVRT method validation or analytical method validation. And with the resources we have currently available, guidance, trainings, I think a majority of those challenges are resolvable within one cycle. With IVPT, however, we see that approximately 79% of the ANDAs that we have pending and which require an IVPT, the IVPT is inadequate. So this is a significant challenge for us. And when it comes to IVPT, we have observed that there are challenges related to method development. And what I mean by that is we observe that there is inadequate optimization, for example, with respect to the receptor solution. It is extremely important during IVPT method development to optimize the receptor solution for a given drug molecule, a given drug product, to ensure that there is sufficient sensitivity, sufficient solubility and stability of the molecule in receptor solution. And it is often not appropriate to just use a receptor solution that is available in the lab and proceed to method validation and pivotal studies. Another example of a challenge that we observe is with sampling frequency. We often see methods that are being submitted where a standard sampling frequency is utilized without any consideration for how that sampling frequency impacts the shape of that permeation profile, and if that sampling frequency allows us to truly compare two products. So one potential option to resolve some of these challenges, because we do recognize there are challenges with IVPT method development, is to potentially discuss with us as you are developing your IVPT method, as you are running into these challenges through the pre-NDA path, and specifically through the product development meetings. The other major challenge that we observe related to IVPT is related to method validation. And there we see challenges related to the selection of the dose, for example, for the IVPT sensitivity study, or the selection of the alternative formulation for the IVPT selectivity study. And in this situation also, we feel like we have guidance out there about how to select the dose. We have presentations out there about how to select a dose that will give you the separation of the profile between your target dose and your selected dose to be able to establish the sensitivity of the IVPT method. But we also recognize that since assessing sensitivity and selectivity is a qualitative assessment at the moment for our guidances, this can lead to some challenges. So again, as you are developing your IVPT method, as you are validating your IVPT method before you proceed to that expensive pivotal IVPT study, If you have questions, we strongly encourage you to interact with us through either the product development program, or in some situations, even the pre-submission program may be appropriate. There are, of course, other issues related to aberrant data in IVPT studies, and given all of these challenges that we are observing, we hope to host a CRCG workshop in March of next year to address, to discuss a lot of these challenges in a lot more detail. So in conclusion, I would like to say that the landscape analysis of the ANDAs in the topical product generally indicate that characterization-based approaches have been predominantly used to support ANDA submissions since GDUFA 2. And overall, not just using characterization-based approaches, we have approved over 175 topical ANDAs since FY18. Among the pending ANDAs, there are challenges related to pharmaceutical quality, labeling, and/or BE. However, for the products where characterization-based BE approaches were used, the predominant challenge appears to be related to the IVPT studies. So we would like to, again, recommend engagement with agency early during product development, as you are developing your methods, as you are validating your methods, so we can resolve some of these challenges and support product approval. So with that, my challenge question: what are the pathways that can be used to interact with agency? A product development meeting, a pre-submission meeting, a post-complete response letter related meeting, controlled correspondences. I would say you can use all of the above, whichever seems most appropriate for your specific question. So with that, I would like to acknowledge my entire team within the Office of Research and Standards, and I would like to invite Dr. Lei Zhang for her presentation related to ICH M13.